that shows the contents of photosynthetic pigments. Chlorophyll A, chlorophyll B decreased under salt stress, while the different foliar applications in singly and in combined form significantly enhanced the chlorophyll A and chlorophyll B contents in both wheat varieties. And the leaf malone D aldehyde contents and total-soluble proteins increased under salt stress in both wheat varieties. While Dilkash 20 show that the contents of malone D aldehyde were lower as compared to MH21. That shows that Dilkash 20 show less damages in membranes as compared to MH21. Total chlorophyll and carotenoid contents decreased under salt stress in both wheat varieties, while the combined selenium and salicylic acid significantly enhanced the total chlorophyll and carotenoid contents in both wheat varieties. Leaf relative membrane permeability and hydrogen peroxides increased in both wheat varieties under salt stress, and however, the foliar application of selenium and salicylic acid in combination decreased the leaf relative membrane permeability and hydrogen peroxide contents in both wheat varieties, and the response of Dilkash 20 was better as compared to MH21 towards the foliar application of selenium and salicylic acid under salt stress and non-stress conditions. The activity of different antioxidant enzymes like ascorbate peroxidase, superoxide dismutase catalase, and glutathione peroxidase increased under salt stress, while the foliar application of selenium and salicylic acid further enhanced the activity of ascorbate peroxidase and significantly boost the activity of peroxidase, catalase, and glutathione peroxidase. The graphical representation and statistical analysis shows that the activity of different antioxidant enzymes significantly increased under salt stress and under different treatments of selenium and salicylic acid in both wheat varieties. Among other biochemical attributes, total proteins, amino acids, protophenolics, flavonoids decreased under salt stress. However, the foliar application of selenium and salicylic acid increased the protein contents, amino acids, and phenolic compounds in both wheat varieties. Statistical analysis shows that of various treatments, the combined application of selenium and salicylic acid significantly increased the total amino acids and the total phenolic contents, total flavonoid contents, and activity of superoxide dismutase in both wheat varieties under salt stress and non-stress conditions. Ionic balance, shoot and root sodium and potassium significantly increased under salt stress. However, the foliar applications of selenium and salicylic acid decreased the root and shoot sodium contents while increased the accumulation of potassium contents in root and shoot. Similarly, sodium to potassium ratio increased, that is under unfavorable conditions, while the application of selenium and salicylic acid restores ionic homeostasis, and of the two wheat varieties, Dilkash 20 showed more pronounced effects and showed better performance than MH21 under salt stress and non-stress conditions. A byplot of wheat varieties under salt stress and non-stress conditions that shows that team 1 is 68.1% shows variance, while the team 2 shows 12.8% variance among different treatments that are shown in different colors and spheres. Cluster analysis, that is heat map of wheat varieties under salt stress and non-stress conditions. Here, the map shows the clustering of two wheat varieties under stress and non-stress conditions and under different treatments of selenium, salicylic acid, and the combined application of selenium and salicylic acids. Here, the different colors like the light sky color and dark blue color show the two stress levels, while the treatment among the three different treatments, the green color is control, golden color is selenium 1 millimolar, white is salicylic acid 1 millimolar, and of selenium plus salicylic acids, and the two varieties that shows red color is of MH21, green color is Dilkash 20 under control condition, and dark blue is Dilkash 20. The map shows that the Dilkash 20 was higher in the potassium to sodium ratio, grain yield components, and the activity of antioxidant enzymes, and the accumulation of different osmoprotectants like total soluble proteins, phenolics, total free amino acids, and choline and glycine ketone contents under control and salt stress conditions. In conclusion, salt stress of 100 millimolar NaCl reduced growth attributes and chlorophyll contents while increased relative membrane permeability. However, combined selenium and salicylic acid application significantly increased growth yield, activity of antioxidant enzymes like ascorbate peroxidase, superoxide dismutase, catalase, and other biochemical attributes including protein, amino acids, phenolics, and flavonoids. Salt stress increased sodium accumulation in root and shoot and oxidative stress markers like malone D aldehyde, hydrogen peroxide in leaf. Overall, selenium and salicylic acid enhanced salinity tolerance by improving physiological biochemical attributes, ionic regulation, and mitigation of membrane damages. Of two wheat varieties, Dilkash 20 performed better than that of MH21 under salt stress or non-stress conditions. Thank you.
Bismillahirrahmanirrahim. Myself is Professor Dr. Saima Masood, working as a chairperson in the Department of Anatomy and Histology at University of Veterinary and Animal Sciences, Lahore. The study of my topic of my study is histopharmacological effects of deep dye needling on myofascial trigger points in skeletal muscles of Mr. rats. So, what is actually myofascial trigger points? Is described as a pain caused by myofascial pain is pain caused by myofascial trigger points in skeletal muscle fibers. It is common in regional musculoskeletal pain syndrome, either above or in conjunction with other pain categories. So, this study was aimed to address the lack of comprehensive data on the histopharmacological effects of deep dye needling on myofascial trigger points in Mr. rats. So, our study could potentially improve treatment protocols for myofascial point pain syndromes and advanced understanding of cellular mechanisms management. It also focused on histopharmacological changes of deep dye needling in skeletal muscle fibers and myofascial trigger points. So what is the trigger point? Actually, the trigger point is made up of multiple contraction knots. Individual contraction knot is muscular fiber, and the fiber segment that has severely contracted sarcomeres and larger diameter. So severe localized sarcomere contraction have been observed in both animals as well as in human beings. Our study was actually in our study 40 male and female blister rats were taken and divided into four groups having age 6 to 7 weeks. First was control group and negative control group. Second was deep dye needling group, and the third was trigger point deep dye needling induction by the neostigmine at the rate of 0.1 induction by kg body weight, and group 4 was per kg body weight, and group 4 was actually having both myofascial trigger points and deep dye needling induction. Both styles of pain was clearly seen with the sign and symptoms. So each needling session consisted of giving approximately 10 to 15 punctures in and out approximately 10 to 15 movements to release the trigger points by vocal switching response. So our results regarding histopharmacological analysis of gastrocnemius muscles indicated how we process the slides that by taking samples of gastrocnemius muscles and then classification in buffered formalin solution and grading using ascending grades of ethanol for dehydration purposes, and later on we did microtomy and after staining with standard hematology and using protocol. We analyzed the muscle sectors under microscope. The muscles here you can see the micrograph indicating different groups, muscle groups and different studies of muscle control group, and A is indicating muscle fiber, while B is muscle fascicle, and C is endomysium, which is a connective tissue there around the muscle fiber, and periphysium along with the connective tissue covering along with the muscle fascicle. So you can see the description of results here that G1 group is actually having normal saline, and G2 group deep dry needling group, and the third is G3 group, which is trigger point group, and fourth group is having both trigger points and deep dry needling. Then histopharmacological analysis of gastrocnemius muscle was done, and not only using HT staining, and not only using HT previous slides were at Lennox, but these slides are at 40 and of length, and these are different histopharmacological
चलो
analysis.
ठीक है आप कोशिश करें कि इसको जितना भी लिखा है कल आके चेक करवा
to indicate the connective tissue or the collagen fibers around the muscle fascicles and around the individual muscle fibers.
हाँ जी
We can see that in third group, this is localized. This greenish color is indicating the collagen fibers in between the muscles, and in the fourth group, you can see that
कितने
this localized collagen fibers, they were moved around they were dispersed at the termination of the recovery phase, just close to the normal. So these are the different scales and by using scales which are used in rats, cat and
तो सही
scale, we
करवाना
can see that how the pain can be relieved, and these are the different facial expressions of the rats during these
हाँ
induction
वो तो अपने
points.
को हैगा
And in first picture you can see that this is normal, and the second picture is telling you the facial points, the facial expressions of the mouse with the dilated eyes, and its ears are at the periphery. And our study is indicative of the that deep dye needling, not only the myofascial trigger points, but this both combination of deep dye needling and myofascial trigger points are significantly controlling. They are significantly contributing to normalization of the morphometric parameters, and they are helpful in decreasing.
मुझे सर पता
They are
किया
helpful
देखो
in decreasing. They are helpful in increasing that pain, myofascial pain, and these all findings are very
ठीक है
obvious histological findings. And we can see that deep dye needling improves the histological parameters and reduces the pain, myofascial trigger points, supporting its potential as well as it is effective for therapeutic approach for the myofascial pain syndrome. Thank you very much.
शहादत दी मैं आ गया
Bismillahirrahmanirrahim. In the name of Allah, the most gracious, the most merciful. Good morning, respected chairperson, all the organizers of this conference, honorable guests, distinguished researchers, and all the audience. I welcome you all to International Brain and Biomedicines Conference in 2026. My name is Seher Gul. a gold medalist in MSc Zoology from Gumal University, and an MPhil scholar from University of Agriculture, DI Khan. It is an honor for me to present my research work here. My research work in MPhil was comparative evaluation of antidiabetic, antioxidant, and antibacterial potential of agaricus leukocytes and Pleurotus aspiratus mushrooms as and their implications for metabolic and oxidative stress related disorders. Specifically for this conference, I will try to align my work
Our study could potentially
with
improve treatment protocols
the
for
objectives of this
myofacial
conference, and will try to tell about the societal, about the health, about the body impacts of these disorders, and also how AI can be used in enhancing the role of mushrooms in managing these disorders. As for diabetes mellitus, we know that diabetes is a metabolic disorder which is characterized by hyperglycemia, chronic hyperglycemia. And according to International Diabetes Federation, about 557 million adults are affected globally because of diabetes. And in Pakistan, it is 26.3% adults which are affected of diabetes. Diabetes has certain complications associated to it, like neuropathy, retinopathy, nephropathy, and it is all because of the oxidative stress which occurs in diabetes. As you know, that people with diabetes have their eyes affected, they have their kidneys affected, they have their brain affected because of this disorder. The traditional therapies are there, like insulin, metformin, but they have certain limitations, they have certain side effects, cost and accessibility problems are there. So there is a need for alternatives, such alternatives which will have which will be multi-target and which will manage diabetes, and not only diabetes but also the antimicrobial resistance. As in Pakistan, we know that we have about 60% of the antibiotics which are not as functional as they should be because of the antimicrobial resistance developed against them. So we are in need of certain alternatives, and these alternatives, and for these alternatives, we look into the nature's pharmacy, and mushrooms are there. For the brain impacts of the oxidative stress of the disorders, certain disorders like diabetes and antimicrobial resistance, we know that as I talked about that diabetes has in diabetes free radicals increase in our body, and these free radicals result in neurodegenerative diseases like people suffer from Alzheimer's along with diabetes. Also, diabetes is linked with cognitive decline, with memory impairment. Also, we know that in diabetes the wounds repair they take time in repairing, or they don't heal properly. Also, it is because of the increased free radicals in our body during diabetes. If we try to get antioxidants into our body, and such compounds, such products, such foods which are rich in antioxidants, then they can manage this neuronal damage, this inflammation, and mushrooms are rich in that. So they support neuroprotection. Also, if you look at the societal impact, I already many people globally, how many people are suffering from diabetes in Pakistan, and how antimicrobial resistance is there, how the traditional medicines are not working because there is a misuse of antibiotics, people are misusing it, and in low and middle income countries, there are not the novel antibiotics are not there because of the cost, because of the there is a lack of accessibility to them. So therefore, we need certain natural and certain affordable alternatives like mushrooms, which will be sustainable and which will be nutraceutical resource. Why I used mushrooms? Because mushrooms are a rich source of polysaccharides, phenolics, flavonoids, terpenoids, and these all compounds have proven antidiabetic, antioxidant, and antibacterial properties, and even anti-cancerous properties. My focus was on two species, as I talked about, Leucogericus leucothides, white mushroom, and Pleurotus aspiratus, or oyster mushroom. For this, review of literature is already available. We suggest that mushrooms are a rich source of antioxidants. They have anti-hyperglycemic effects. They can manage oxidative stress. They have anti-inflammatory properties, and they can be used as interventions for diabetes. My problem statement was that no studies directly compared these two species simultaneously. They are antidiabetic, they are antibacterial, and antioxidant effects, leaving their therapeutic potential unclear. This critical knowledge gap hinders optimal use of these mushrooms in diabetes and infection management. My hypothesis was Pleurotus aspiratus exhibits a higher antidiabetic potency, antibacterial, and antioxidant property than Leucogericus leucothides. My aims and objectives are to evaluate and compare the antidiabetic potential of these mushrooms, to assess their antibacterial activity, and to investigate and compare their antioxidant activity also. Methodology which I used was I collected mushrooms, I prepared their extract, I then performed the antibacterial activity using this diffusion method, antidiabetic activity using alpha amylase inhibition, and antioxidant activity using DPPH assay. As per my methodology, I collected these mushrooms from Paharpur, DI Khan. I get them authenticated from a mycologist. I dried them. I powdered, made a powder of them, and then through microwave assisted extraction, I prepared their extract. The methodology, as I stated, that alpha amylase inhibition using iodine starch method was used for anti-diabetic assay, for antibacterial test, this diffusion method, antioxidant for antioxidant activity, DPPH radical scavenging was used, and then statistical analysis was done. These are my results, which show that the Pleurotus aspiratus showed a greater alpha amylase inhibition. It inhibited alpha amylase activity more as compared to Leucogericus leucothides. As for the antioxidant activity, the ascorbic acid, which was used as a standard antioxidant, showed the best results. Then came the Pleurotus aspiratus, and then the Leucogericus leucothides, which showed middle activity. These results are shown here. As you can see, that ascorbic acid has a best antioxidant activity, Pleurotus aspiratus has medium, and then Leucogericus leucothides. As for the antibacterial activity, I used three strains of bacteria, S. aureus, gram-positive bacteria, against which both of the mushroom species showed very good results and larger inhibition zones as compared to these mushroom species against Escherichia coli and P. aeruginosa. It may be probably due to the cell wall structure. There is a literature gap. If someone wants, they can dig into that. So for discussion, we know that Pleurotus aspiratus and leucothides have showed greater amylase inhibition, but stronger inhibition was showed by Pleurotus aspiratus. It is because it is rich in flavonoids, it is rich in phenolic compounds, and these compounds inhibit alpha amylase activity. Also, the antioxidant activity of both extracts was there, but Pleurotus aspiratus showed greater because it is rich in phenolic contents. Moreover, antibacterial activity against S. aureus was greater as compared to Escherichia coli and P. aeruginosa, and it is also due to the presence of antibacterial compounds, glucans, phenols, etc. Then we have the conclusion. As we stated in our hypothesis, that Pleurotus aspiratus showed greater activity, so our hypothesis was proven true, that it has greater activity, it has significant antidiabetic, antioxidant, and antimicrobial activity, and it is due to those compounds which are present in mushrooms, like phenols, like polypeptides, like we have the glucans, these compounds, and these are effective at managing diabetes, infections, and oxidative stress. For the use of AI, we can use AI in isolation and identification of bioactive compounds from mushrooms. We can use it for in vivo validation, for dosage optimization, and machine learning models of AI can predict antidiabetic and antimicrobial activity of bioactive compounds. They can also stimulate greater interaction between mushroom compounds and enzymes like alpha amylase. And these compounds, if they have a greater interaction, they will show a greater alpha amylase inhibition activity. Then we will, they can also, AI models can also be used for in silico testing before we go for in vitro validation. Moreover, novel compounds from medicinal mushrooms can be isolated faster because of this. And also, AI can be used for anti-cancerous, anti-diabetic compounds for their discovery, and it can screen bioactive molecules faster than lab testing, because in lab testing we will have to look at certain, we will have limited chances for looking into compounds, for looking into their bioactivities. But for if we develop certain AI models, then we will have greater chances to dig into those compounds which have therapeutic potential for these disorders. And also, AI models can analyze mushroom metabolites, which will be used for against Escherichia coli, against certain specific bacteria before we can conduct lab experiments. I mean that there is no need for conducting lab experiments if we have certain models of AI in our hand. At last, I will thank you all for your kind attention. I look forward to your questions and very.
Assalamu alaikum everyone. I'm Zirina Ikram, and today I'm presenting my topic that is enhancing nutritional status. First of all, introduction. Poor dietary habits are increasing globally. They're linked with obesity and malnutrition and chronic disease. Eating behaviors plays a critical role in health. Traditional nutrition approach focused on what to eat, but less attention is given to how we eat. So mindful eating addresses this by targeting eating behaviors and awarenesses. So unhealthy eating patterns such as overeating, emotional eating, irregular meals are becoming more common. So these behaviors contribute to both undernutrition and overnutrition. But mindful eating has emerged as a strategy to improve awareness and promote healthier eating habits. Next is, what is mindful eating? So let's understand what is mindful eating. Mindful eating means you are conscious awareness during eating, focused on your present moment, recognizing your hunger and satiety signals, eating without distraction, non-judgmental attitude. Mindful eating means being fully aware of what and how we eat. It focuses on experiencing food through our senses and recognizing hunger and fullness signals. This approach helps us improve control our eating habits, reduce our emotion and mindless eating, and support a healthier lifestyle. Okay. So mindful eating involves multiple components. It enhances our sensory experiences, our taste, smell, and it also helps manage our emotions. So celebrate food choices, promote eating, which improve digestion and satiety. When people practicing mindfulness, they become more aware of their emotions and the reason behind their actions. The awareness helps them make better choices, like not turning to food every time they feel stressed or sad or upset. In this way, mindfulness acts as a helpful tool for self-control, allowing people to manage their emotions instead of reacting automatically to emotional eating. Next is mindful versus mindless eating. So let's understand what mindful eating is and what is mindless eating. In mindful eating, a person eats slow, have attentive eating based on hunger cues, not on emotional driven eating. Also, they have controlled portion. They take control portion, you can say that. But in mindless eating, they eat fast, distracted eating, emotion driven eating, and which can lead to overconsumption. Mindless eating often occurs when individuals eat while distracted or stressed, which lead to overeating. But in contrast, in mindful eating, it promotes control and awareness, reducing unhealthy habits of eating. What is the role of mindful eating with nutritional status? Yeah, what is the link? So, improve dietary quality. Mindful eating improves dietary quality. It also enhances nutrition intake and prevent overeating, support healthy body weight, improves digestion. Mindful eating help individuals consume balanced diet and maintain a healthy weight, which is essential for overall health. Studies shows that it helps reduce belly fat and improve blood sugar control in overweight individuals. So these things shows that mindful eating has a greater impact on our nutritional status. These benefits shows that mindful eating supports better nutritional status by improving both eating habits and body functions. Okay, so mindful eating role in emotional eating. As mindful eating increases, emotional eating decreases. You are conscious aware of what you are eating. So mindful eating improves emotional control, help identify the true hunger, reduce the cravings. In emotional eating, you eat, binge, but mindful eating helps to control emotional eating. Emotional eating is common, especially among the students and stressed individual. Mindful eating helps individuals distinguish between emotional and physical hunger and reducing unnecessary calorie intake. When the literature is examined, parallel to the findings is found, it is known that regular practices of mindfulness practice facilitate the acceptance and tolerance of negative emotions. So the scientific evidence shows that mindful eating is associated with lower BMI. It shows that a person who do mindful eating has lower BMI, reduce binge and emotional eating, improve diet quality, support long-term behavior changes, and research studies shows that individuals practicing mindful eating tend to have better dietary patterns and reduce emotional eating. It is considered an effective and long-term strategy. So here are some practical strategies you can apply in your life to do mindful eating. First is eat slowly and chew thoroughly. Second, to avoid distraction. No TV, no mobile while eating. Listen to your hunger and fullness signals while eating and practicing your portion control. That's the most important point. And keeps a food journal in which you can write your eating patterns, your eating habits. You can improve day by day. Simple strategies can help implement mindful eating in daily life. So the conclusion shows that mindful eating improves eating behavior. It also enhances nutritional status. It reduces emotional eating, and also it is an easy and practical approach. In conclusion, mindful eating is a simple yet powerful approach that can improve both behavior and nutritional outcome. So it should be promoted in public health and clinical settings so that more people are aware of what is mindful eating and to practice of mindfulness in their life so they can have more benefits of it to improve nutritional status, less emotional eating, which can lead to a healthy life. So these are the references. Thank you so much.
Assalamualaikum. I am Sudhakar PhD scholar at the College of Food Science and Technology, Guangdong Ocean University, China. Today I will be presenting my research on how astaxanthin help preserve the quality of chicken meat under stress by letting antioxidative and anti-inflammatory effects. First of all, start with the background. Global warming is threatened to all kinds of life on Earth, especially the alien species which are highly susceptible to stress. Stress occurs when the amount of food produced by animal exceeds than its capacity to display to the external environment. Broilers are more susceptible to stress due to their thermoregulatory system and faster growth. This stress can be categorized into acute and chronic depending on the intensity and duration of heat. Both acute and chronic stress is harmful to the production and welfare of animals. This stress results in decreased growth rate due to the reduction in feed intake of birds. Chronic stress has a long-lasting effect on broiler as it changes the protein and fat metabolism at the molecular level. Astaxanthin is a carotenoid polycarbonate compounds that have strong antioxidative activity. Its antioxidant activity is 100 times greater than vitamin E and beta carotene. Due to its high antioxidative activity, it can be used as a feed ingredients to mitigate the heat stress effects. Research methodology. In our study, we tested the effect of astaxanthin supplementation on broiler exposed to the heat stress. We divided the broiler into 5 groups. Group A was control group, Group B was the heat stress group, Group C, D, and E was the heat stress group, and supplemented with 20 milligram, 40 milligram, and 80 milligram per kg of astaxanthin. And after the 14 day of its test, we have taken the 5 muscle samples for meat quality analysis, and also we have collected the blood for immune status determination. Results. Physical meat quality analysis was done after the 14 day of stress, and we observed that there was no difference in pH, water holding capacity, weight loss among the group after chronic stress. The redness value and the yellowness value were significantly different, and the group D had the highest redness value. Cook loss in the group B show was significantly higher than the other groups, and the share force of the group B was significantly higher than the other groups, but there is no significant difference observed in other groups. Texture profile analysis showed that there was no significant difference in texture profile parameters. Lipid oxidation test was done by using the MDA test that was done by ELISA. The level of MDA in group B was highest to a significant oxidative stress response. The MDA level in group C, D, and E decreased significantly, indicated that astaxanthin have dose-dependent antioxidative effect, and it can effectively reduce lipid oxidation damage caused by stress. Antibody titer was determined by using the
Easy and
ELISA
practical
level of
approach.
IgA, IgM,
In
and
conclusion,
IgY.
mindful eating is simple yet powerful approach that can improve both
than those
behavior
of
and
stress
nutritional
group.
outcome. So it should be promoted.
Muscle fiber analysis show that this stress significantly reduced the muscle fiber cross-sectional area, muscle fiber diameter, and muscle perimeter compared with the control groups. And the difference reached a significant level among the group A versus B. Although the astaxanthin supplementation showed the recovery trend, but it did not reach the statistical significance compared with the group B. Gene expression analysis was done by using the 2 PCR on the 1 level, and it showed that astaxanthin significantly upregulated the antioxidative genes such as estrogen, catalase, and GPX in both muscle and liver tissue that show the enhanced oxidative defensive system. The hystress group showed the increased level of lipogenesis-related genes such as FAST and ACCA, indicating the fat accumulation, but astaxanthin normalized these levels. Additionally, the astaxanthin downregulates the stress and inflammation-related genes such as HSP70, NFK beta, and TLR4, indicating its anti-inflammatory response. Conclusion: astaxanthin acts as a strong antioxidant agent and helps to reduce the oxidative stress and lipoxidation caused by the heat stress. The astaxanthin at the rate of 40 milligram per kg showed the most significant improvement in both meat quality and the immune response. These findings suggest that astaxanthin could be an effective supplement to improve the quality of chicken meat exposed to heat stress conditions. Thank you all for your attention. I am now happy to take any question you may have.
Assalamualaikum. It's me, Yamsha Jabbar, special graduate student from Psychophysiology Research Unit, Department of Physiology, University of Karachi. And my presentation topic is better nights, brighter days, sleep hygiene and aromatherapy in combating daytime sleepiness. Let's begin the presentation with a simple question. Have you ever felt sleepy despite having a good day? Good morning. We all know sleep is vital for our cognitive functioning. But for young adults, it's often the first thing sacrificed to academic stress and late night scrolling. Our study, Better Nights, Brighter Days, explores how we can fix it through the simple non-pharmacological approaches, which are sleep hygiene and lavender aromatherapy. So, what does it actually mean? How does it work? Let's break it down. Sleep hygiene means creating a bedroom environment that is comfortable with limited disruptions. Whereas aromatherapy works by stimulating the olfactory system, which calms the nervous system and lowers sympathetic arousal. Sympathetic arousal required are stress periods. Let's talk about the methodology, that how the study has been done. We conducted a 4-week randomized control trial with young adults ages 18 to 25. We didn't just ask them how they felt. We used a double check method by collecting both subjective and objective data. What does subjective and objective data means? The subjective data is the data which the individual report directly to us, while the objective data is the data which was collected indirectly from the individuals. So how the subjective data was collected? In subjective data, we used standardized scales like Pittsburgh Sleep Quality Index (PSQI) to check out the sleep quality, sleep hygiene index to monitor their sleep hygiene, and upward sleepiness scale (ESS) to monitor the degree of severity of their daytime sleepiness. Whereas objective data was collected through the Sleep Cycle app, which is actually a smartphone app, in which we have monitored their sleep latency period, time of awakening, time going to bed, etc. So we actually selected individuals who were having high scores of PSQI, SHI, and ESS and randomly assigned them into two groups, which were interventional group and control group. The interventional group has to follow sleep hygiene practices like following fixed bedtime, no screens before bed, and calm room. Whereas the control group was free to do whatever they want. The individuals who were following the aromatherapy as an intervention were asked to use lavender oil 30 minutes before going to sleep each night. So that's how the study has been done and the data was monitored. So let's move towards the most amazing part of this study, which are results. This is the data. The baseline mean is the mean that was monitored before starting any intervention from the individuals. So the ESS score was 15.55, reported by the individuals, which is actually the mean of all that scores. So after following the intervention, they reported 3.36 score, which here you can see how much it has been dropped down. Here the PSQI was 16.5 reported before following any intervention, and after following intervention, it dropped down to 9.27. SSI was 28.8, and after following intervention, it turned into 9.88. So here you can see how magically our study worked. Not only these things, but here you can observe the p-value. The p-values. The p-value is lesser than 0.0001, shows this wasn't just a duck. The changes were highly significant. Let's move towards the results of aromatherapy. Here, the individuals who followed aromatherapy first reported before following any intervention, the mean score of PSQI was 8.47. And after following intervention, it turned into 3.76. While the SSI score was 34.26, whereas after following intervention, it turned into 27.23. Here you can see 3.76. PSQI score actually lesser than 5 lies into this category of good sleep quality. Here you can see individuals reported 3.76 mean, which actually lies in the good quality sleep. While the control group, who didn't follow any intervention, reported it 8.63. 8.47 was worsened in the control group. Here the SSI score, we can see it dropped to the 27.23, whereas in control group it comes down to 31.96. Here you can see there is a big difference between these two values, but there is a small difference. So intervention, which is an aromatherapy, actually works on the individual who were facing these sleep quality issues. Let's move towards the objective data. Objectively, the interventional group gained an extra 71 minutes of actual sleep per night. They were not just staying on that longer, but they were sleeping more efficiently. We saw a 130-minute increase in time spent in bed, meaning participants began prioritizing their sleep. Most importantly, these were statements suggesting that this isn't just a short-term fix, but a sustainable habit for long-term mental and physical health. So these results prove that actually our sleep hygiene and limited aromatherapy work on the individuals who were facing sleep issues. Let's conclude our presentation. There is no need to do any complex medical intervention to fix your sleep issues. We only need a drop of lavender oil and some good habits to make us healthy and productive. Hope you like my presentation. Thank you so much.
Assalamu alaikum everyone. My name is Dr. Evelyn Sabha, and I am serving as Assistant Professor for Veterinary Physiology in the Department of Veterinary Biomedical Sciences, Faculty of Veterinary and Animal Sciences, P. Meher Ali Shah, Agriculture, University, Rawalpindi. It's a privilege for me to present the research from my lab in this prestigious conference, that is International Brain and Biomedicine Conference. Today, the topic which I will be discussing with all of you and sharing my research, that is the anti-inflammatory activities of cinnamon, henna, and turmeric in chicken peritoneal macrophage. Even though that this topic really does not relate with that of the brain, however, because the theme included veterinary sciences and medicine, so therefore I'm here to present my work on this topic. So the main focus of the research is actually inflammation. Inflammation, most of you may know that it is a biological response which is triggered by many factors. In fact, it is actually the natural response of body towards a foreign invader. And it is characterized by 5 cardinal signs, which we mostly say, that is redness, loss of function, pain, swelling, and heat. So there can be 2 types of inflammation, one which is short-lived, that is acute, and the other one which may have a long time, that is chronic inflammation. So for the treatment of inflammation, normally there are 2 sets of drugs which are mostly used. That is steroidal anti-inflammatory drugs that are called as SEDs, and non-steroidal anti-inflammatory drugs that are called as NSAIDs. And here in the table you can see that there are different set of drugs which are actually available over here for the treatment of inflammation. Now, given that there are different set of treatments which are actually available for the treatment of inflammation, that is the SEDs and NSAIDs, even though that they have lots of effects which are good, that is why they are being used, that is anti-inflammatory and analgesic and anti-arthritic and so on. However, dependence on these drugs, which happens most of the time, may lead to a lot of
side effects like hepatic and GIT dysfunctions, cardiovascular, and even cerebral complications. So, what is actually solution for that? How we can actually use something which will be anti-inflammatory as well as it may not also give lots of side effects? Because having said that, inflammation is actually going on in a very quick and almost every time in the body. So, dependence on SERDs and NSAIDs is very natural, but there can be a shift towards the alternative therapy to reduce the use of these SEDs and NSAIDs. And having said that, there has been a lot of shift towards the alternative or the herbal therapy since past lots of years. Why? Because they are mostly non-toxic, even though that there are some compounds in these things which are toxic. However, broadly, they are almost non-toxic. Plus, they are easily available and also they can be sourced locally. In fact, I would like to say here that most of the allopathic drugs or the synthetic analogs that we use, actually they are the copies, or you can say they are the chemical alternatives of those single compounds which are present in these drugs. So, in my research, I selected three commonly available herbs and spices, that is turmeric, henna, and cinnamon, because all these three are very much locally used in our culture and in our country. So it is very easy to procure these compounds and they can be likewise used. So turmeric, as we all know, Urdu mein isko haldi kehte hain, and the scientific name is curcuma longa. And everyone already knows that it is having lots of anti-inflammatory effects. The second one was cinnamon. Isko Urdu mein darchini bhi kaha jata hai, and this is also a very commonly used spice, and also it has reported potent anti-inflammatory effects. As when we come to henna, henna ko Urdu mein mehndi bhi kaha jata hai, and this is mostly applied for dyeing purposes and also for cosmetic purposes. However, in the pure form, henna has got lots of anti-inflammatory effects. Now, why I use these ones, as I told before, that they are very much locally available, and I actually wanted to check their effects in the inflammatory model, and for that reason, I actually used chicken peritoneal macrophages. So, material and methods included all of these, as you can see over here. A sample was collected, and their extracts were prepared, birds were procured, and sheep red blood cells were prepared, hyperimmune anti-serum was prepared, then the macrophages were isolated, and then they were given the sheep red blood cells, and we checked their phagocytic activity. Then we also checked either these extracts, did they show any kind of cytotoxicity, and for the anti-inflammatory effects, we checked the NO assay. And we also check the expression of genes which are related with the inflammatory pathway. Finally, for the significance, we used statistical analysis. So what the results of this study showed, as you can see here, that henna, cinnamon, and turmeric, all three of them, actually we used two doses of both of all three of these herbs and spices. So the highest doses, they phagocytosed most of the sheep red blood cells. It showed that these extracts actually, they enhanced the phagocytic activity of the macrophage. I mean, normally macrophage do phagocytose. That is a part and parcel of their function. However, when we treated these cells with the different spice extracts, their phagocytic activity was increased. The arrow in these pictures is actually showing the RBC, and the other remaining cells, you can say they are the macrophages. And for the cytotoxicity, we did not find any cytotoxicity for these extracts, as it is shown by this MTT assay for various doses. And for the nitric oxide production, because it is nitric oxide is naturally produced as a result of inflammation. However, its overproduction may lead to the signaling of the inflammatory pathway. So for any extract or anything that has to show its anti-inflammatory activity, it should decrease the NO. So what we find here is that the extracts at different doses, they decrease the NO activity. And further we move towards the expression of inflammatory mediators and cytokines. So INOS and FOX2 are the mediators, whereas TNF alpha, interleukin 1, beta, and 6 are cytokines. So as you can see that all mediators and cytokines also were decreased with the treatment of these extracts. And finally, we found out the pathways which are actually involved for the inflammation. So there are two pathways, that is NF-kappa B and MAPK. So they are the classical pathways of inflammation as well. So we can see in these graphs that these extracts not only decrease the expression of NF-kappa B, but also the different subfactors of the MAPK, that is ERK, JNK, and P38. So overall, we can say that actually the inflammation was induced by the classical NF-kappa B and MAPK pathways, and it was reduced by these extracts. So as you can see here, that there was a good anti-inflammatory activity. But in our study, we found that turmeric was actually very much potent. Among all three of them were good. However, turmeric was most potent. So what we suggest is that in order to bring these spices for animal use, there has to be in vivo trials. And further, later on, there can be formulation of different drugs or different kind of treatments for use in the veterinary. So that is it from my side. And these herbs and spices overall, they're very good for inflammation overall. And not only for, you can say for peritoneal, but for the overall inflammatory conditions in the body. So they can be very much inculcated in the normal diets of animal as well as the human beings. Thank you very much.
Bismillahirrahmanirrahim. Assalamu alaikum everyone. I am Dr. Mariam from Department of Pharmacology, Faculty of Pharmaceutical Science, University Faisalabad. Today I am presenting my study on anti-inflammatory potential of Dermatopsis musculata methanolic extract in an experimental polyarthritis model. First of all, there
यार
is
इसको
a
बुला के ये सही करवा
polyarthritis is a
देना
chronic inflammatory disease associated with pain, swelling, and bone degradation. Current therapies are effective, but mostly associated with severe side effects. So there is an urgent need to develop therapies to manage the problem state. Compared to the synthetic drugs, plant-based medicine or therapy cause fewer or less side effects. Research gap. The plant of the study is Termiopsis musculata, which is also called African costa, belongs to the family Astraceae. Despite its traditional use, scientific evidence on its anti-inflammatory, anti-polyarthritic effects are limited. The phytochemical profile of the plant has not fully characterized. No systematic in vitro and in vivo studies have validated its folkloric use. The molecular mechanism underlying its activity is still unclear. The study aimed to bridge these gaps through detailed scientific and molecular evidence. Aim of the study. The aim of the study was to evaluate the anti-inflammatory and anti-polyarthritic potential of Termiopsis musculata, methanol extract in formaldehyde-induced polyarthritic rat. Then there is the experimental design. Phytochemical analysis was performed prior to the detailed in vivo studies. Animal model was formaldehyde was the chemical which was used to reduce the polyarthritis in the rat. 6 groups were formed. The groups were named as control group, disease control, standard control, and rest of the three were the treatment group, in which we use the 200, 400, and 600 mg per kg dose of the extract. Parameters which were determined or evaluated during the study and at the end of the study were clinical, biochemical, hematological, radiological, histological, and molecular parameters. The first one is the result of phytochemical analysis in which we performed HPLC and GCMS. HPLC stands for high pressure liquid chromatography. It is a quantitative analysis in which we determine the quantities of different compounds. In my plant extract, HP acid, rutin benzoic acid, and quercetin was identified. The HP acid was found in highest concentration. The results are shown in the chromatogram of HPLC. Next are the result of GCMS. GCMS is gas chromatography, mass spectrometry. Here you can see the different peaks. 17 different peaks of the compounds were identified. The lowest peak was determined at 11.07 minutes, while the highest peak was determined or noticed at 13.701 minutes. The results of GCMS are shown in the table form. In the table, you can see the peak number, retention time in minutes, compound name, identified gas number, and area percentage. Clinical outcomes of my study. The overall clinical outcomes are the physical parameters in which we determined or evaluated the results of bar diameter, body weight, and arthritic index. Arthritic index shows the severity of the disease. Here is the graph of the results of bar diameter evaluation. As you can see that the reduction in bar diameter was observed after treating the animals with the extract, plant extract. The results were dose dependent, which means that highest dose gives the best result. Next are the results of arthritic index. Arthritic index of the control group was normal. No inflammation was noticed in the control group, while the administration of standard and plant extract to the animal significantly reduced the arthritic scoring. Next are the results of effect of plant extract on body weight. As you can see that there was a significant reduction in the body weight of disease group, while the body weight was normally increasing in the treatment group. The highest increase in the body weight was noticed in the highest dose group. Next are the results of hematological and biochemical parameters. The first one is the result of HB hemoglobin level. The hemoglobin level was significantly reduced in the disease group while it was improving in the treatment group. Other parameters were LFTs and RFTs, and they were improved in the treatment group as compared to the disease group. The results were mostly dose dependent. Along with the LFTs, RFTs, and blood parameters, we also determined the levels of CRP and rheumatoid factor. CRP level was significantly high in the disease group, which showed the inflammation or infection in the animals, and similarly, rheumatoid factor value was 36.70, which was highest in the compared to the rest of the group, which means that the rheumatoid was present in the animals. Next are the results of radiological examination. Treatment group showed the reduced inflammation and preserved cartilage, while in the disease group, the inflammation was highest, along with the degradation of joints and bones. Next are the results of histopathology. The left side, which is the A side, which shows the ankle tissue exhibiting the reduced synovial hyperplasia and lower infiltration of the inflammatory cells in the treated animals. While on the right side, there is a B picture, which shows the bar tissue showing the cartilage protection in the treated animals. Next are the results of ELISA studies. ELISA studies were performed on pro-inflammatory and HSP marker. HSP70 is a heat shock protein marker, which was significantly high in the disease group, similar to the VNF-alpha and IL-6. The extract suppressed the pro-inflammatory cytokines significantly. The results are shown in the form of graph. Next are the results of RT-PCR studies. The gene expression was analyzed through the RT-PCR studies. Pro-inflammatory markers such as TNF-alpha COX2, IL beta, and NF kappa B were downregulated, while the anti-inflammatory genes such as IL4 and IK beta were upregulated. The results are shown in the form of graphs. Next are the results of antioxidant enzyme acid. SOD and CAT levels were significantly increased in the treatment group, while on the other side, MDA level was reduced significantly, indicating the lower lipid peroxidation. The extract provides a protection against the oxidative stress. As you can see that the CAT level and SOD level was reduced in the disease group, while these are improved or were improved in the treatment group. While on the other side, the MDA level was significantly or considerably high in the disease group, and it was reduced or less in the treatment group. Conclusion of my study: the myopsis musculata methanol extract shows a strong anti-inflammatory activity, acts via the suppression of inflammatory mediators, antioxidant mechanisms, gene regulation, and supports the traditional use of the plant as anti-inflammatory and anti-polyarthritic agent. These are the references which I have used during the study. Thank you so much. Allah Hafiz.
Assalamualaikum. First of all, I would like to thank the organizers for providing me this opportunity to present my research at this international forum. I'm Saliha Kramat from the Department of Pharmaceutics at Government College University, Faisalabad. And today I will be presenting my work on the topic Design and Evolution of Silica-based Solid Dispersion Containing Naproxen for Rheumatoid Arthritis. On focusing on this, I explain its objective, its problem, methodology, and its significant finding. So let's begin. So the topic of my presentation is the design and evolution of silica-based solid dispersion containing naproxen for rheumatoid arthritis. The contents of my presentation is the introduction, its aim and objective, problem, methods, and its results. And conclusion. As for this, many drugs have low water solubility, which make them difficult to formulate into the effective dosage form. Because of this, high dosage is required to achieve its therapeutic effect on the body. Therefore, improving the resolution rate of such drug is very important as it can enhances the oral bioavailability and also reduces the dose required to produce its therapeutic effect. One effective approach to overcome this problem is the solid dispersion. In this method, drug is dispersed in a non-reactive carrier as in the solid dosage form. In this solid dosage form, that will increase its solubility. Several studies demonstrated that the solid dispersion is very important or enhances the solubility and bioavailability of such poorly water insoluble drugs. So this approach plays an important role in the improving drug performance. So the aim and objective of my presentation is that to develop the ternary solid dispersion of naproxen using polymers with the non-order mesoporous silica that will improve its solubility, bioavailability, and also reduces the gastrointestinal injuries, and to develop the binary solid complexes with the different grades of silica such as solid 244Fp, solid AL1Fp, and solid XTP3150. And to develop the ternary solid dispersions with the different polymers like polexamer 188, Udragate L100 with the non-ordered mesoporous silica of solid 244Fp. I use solid 244Fp as it increases the solubility as compared with the other grades of the silica. And next is the problem statement of the drug. The problem by which I study this is that naproxen is the non-NSAID that is from the propanolic acid. Is used to treat the dysmenorrhea and rheumatoid arthritis. But the prolonged use of the naproxen may cause the gastrointestinal injuries and the inflammation into the joints. Naproxen belongs to the family of BCS class 2 system. Drugs having these are poor aqueous solubility and high permeability. My innovation of this study is that to improve the naproxen solubility and bioavailability in order to produce the strong anti-inflammatory effect and to reduce the gastrointestinal injuries. Next is the methodology of the binary solid dispersion, in which I dissolved the naproxen into the ethanol, as it is insoluble into the water, and then placed it into the glass pestilent mortar and added the solid. At 300 rpm and 50 degrees centigrade, I placed it into the magnetic stirrer for 1 hour. After 1 hour, I removed it from the magnetic stirrer and placed it into the hot oven and for overnight drying. After that, I collected its powder. Then the general methodology for the ternary solid dispersion is that I dissolved the naproxen into this ethanol and added my polymers like polexamer 188 and Judragate L100. And by its complete dissolution, I added RPM and 50 degrees centigrade. And after 1 hour, I removed it from the magnetic stirrer and placed it into the hot oven, air dryer for overnight drying. And after that, I collected its powder. Then the composition of the binary solid dispersions, I used 3 different grades of silica with the different concentration. My formulation F1 solid 244 1:1 is used as the optimized formulation as the solubility of this formulation is maximum as compared to the other derivatives of this. Then the composition of ternary solid dispersion, in which my formulation F13 and F17 shows the greater solubility as compared to the other derivatives of this. Then the results are entrapment efficacy and the solubility of the binary solid dispersion, in which in the solubility in distilled water pH 1.2 and pH 6.8, the solubility increases, and the entrapment efficacy is also maximum into the formulation F1. Next is that the entrapment efficacy and the solubility of ternary solid dispersion, in which my formulations F13 and F17 also shows the solubility in distilled water and pH 1.2 buffer and 6.8 buffer, solubility increases and the entrapment efficacy is also increases. That shows in which I characterize these formulations and for further studies. Next is that the in vitro dissolution studies at pH 1.2. As time passes, the release of the drug in the formulation in my formulation F1, F13, and F17, the release of the drug is more as compared to the pure drug. And in vitro dissolution studies at pH 6.8, it also shows us that as time passes, the release of drug in formulations F1, F13, and F17 is more than that of the pure drug. Then the characterization FT shows that the pure drug absorbance and the formulation F1, F13, and F17 absorbance, there is not much differences. It shows that the chemical nature of the drug is not denatured, and the slightly change in the absorbance shows that there is a hydrogen bonding between these that can prevent it from decrystallization. The next is the SEM analysis. In this, then pure naproxen shows the smooth and crystalline structures and the formulation F1, F13, and F17 shows the shorter and irregular structures. That shows us that the pores in which the naproxen is embedded into the pores of the silica xylite. Next is the evaluation the effect of gastroprotective in vivo. In this, the control group shows no gastric ulcers as there is no injection of pure naproxen or any other formulation. In pure naproxen F1 formulation and F13 and F17 formulation shows that the circle shows there is a gastric lesion into the stomach, and my formulation F13 and F17 shows is less gastric lesion that can shows that there is the effect into the formulations. Next is the gastric lesion, as the F13 and F17 shows less gastric lesion as compared to the pure naproxen. Next is the evolution of anti-inflammatory activity. It shows that the anti-inflammatory inhibition is reduces into the formulations F13 and F17. That shows its effect. And there is a 6 or photographs of the paw of rats that shows that the inflammation reduces as time passes. The interleukin, serum interleukin 6, that is the biomarker that shows us that the disease group is produces or induce the inflammation. And in the formulation F17 and F13 shows us that the inflammation is reduces. In TNF alpha measurement, it also shows us that the inflammation is reduces in the formulation F13 and F17. And conclusion, I use two carriers to utilize to improve the solubility, dissolution rate, and bioavailability of the drug naproxen. TSD is ternary solid dispersion shows more or enhanced bioprotective and anti-inflammatory effect as compared to the DSD. So the polexamer 188 and Judragit 800 are used to reduce the stomach irritation that caused by the NSAIDs. Thank you very much for your time and attention. It has been a great honor to present my work here. I truly appreciate the support of my supervisor and the opportunity provided by the organizer. I look forward to your queries. Thank you.
Assalamu alaikum. My name is Mohammad Hasnath. Today I will present my topic triptolide and mitochondrial toxicity. These are the actually it is hepatotoxicity, and first of all there is abstract. The objective of our study was to find out the link between mitochondrial dynamics and cell death in triptolide-induced hepatotoxicity. Triptolide, it is active ingredient of Chinese herbal plant and it causes hepatotoxicity. So in this study, we evaluated the hepatotoxicity caused by triptolide. We treated LO2 cells with 25 nanomolar concentration of triptolide. The results demonstrated that triptolide treatment caused an increase in apoptotic cell death, mitochondrial depolarization, ROS overproduction, decrease in ATP production, and mitochondrial fragmentation, which causes the activation of DRP1 fission protein. Triptolide treatment led to the translocation of DRP1 from cytosol into the mitochondrial membrane, where it causes the mitochondrial fission. Transmission electron microscopic analysis revealed that there is distortion of mitochondrial morphology in triptolide treated group. Western blot analysis also explained that there is recruitment of DRP1 to mitochondria, cytochrome C release, and caspase 3 activation. However, inhibitor of DRP1, MDB1, inhibited the activation of DRP1 and caspase 3 and blocked the. Here is the results and discussion. Here you can see this is the post-cytometric results. This is the apoptosis results. As compared to control, there is increase in apoptosis in a time-dependent manner because there is the concentration is same. It is 25 nanomolar, but time is being increased from 12 to 24 hours and then 36 hours. Similarly, mitochondrial depolarization also increases as we increase the time. And ROS production, you can also see that there is increase in ROS production. Here is the Western blot results. You can see that this 0 R 3 R 6 R 12 R and 18 R shows that the concentration of triploid it is the dose of triploid is 25 nanomolar. But here you can see that DRP1 level it is increasing or it is constantly increased as we have increased the time, like from 3 hours to 18 hours, DRP1 level is increased as compared to the control group. Similarly, FIS1, there is little bit change in the level of the fission 1 protein. Then there is MFN2, OPA1 and MFN1. These all are mitochondrial fusion proteins. MFN1 level is decreased as we increase the time, and remaining others, there is little bit change or there is no change. As you can see that in OPA1, there is little bit change in the 3 or 6 hour time period, but in other time periods there is no change. But this Western blot result shows that there is increase in the DRP1 level. DRP1 protein, which is our main protein, on which our study is focused, its level is increased. Here you can see that here caspase 3, its level is increased as there is the time is increasing. At 0 hour, its level is low, but as the time is increasing, its level is increasing. At 3 hour, it is much more as compared to the 6 hour, 12 hour, or 18 hour. Then there is mitochondrial fraction. Cytochrome C level as compared to control at 0 hour, there is increase in mitochondrial fraction of cytochrome C, but as the time is increasing, mitochondrial fraction of cytochrome C is decreasing. And DRP1 level, its level is increasing in the mitochondria. So this here we have treated the cell line with control group triploid plus MDB1 and MDB1. As you can see that DRP1 level of mitochondrial fraction is increasing triploid group. And here you can see that caspase level is also increasing triploid group, which is decreased in the TP plus MDB1 group, which means that MDB1 has cured the cells that were affected by the triploid hepatotoxicity. Here you can see the mitochondrial analysis of transmission electron microscopic analysis of mitochondria as compared to control. You can see in triploid treatment group there is severe toxicity. Here you can see the organelles and the cell inside surface it is blank, which means that there is significant toxicity caused by the triploid group and triploid also cause bacterial formation in the mitochondria. If there is bacterial formation in the mitochondria, which shows that there is apoptosis in the mitochondria, and the crystal is also absent in the mitochondria, which is caused by MDB1 treatment. Now here you can see tunnel analysis, tunnel assay, there is apoptosis in the animal tissue of triploid treatment show. And here is the Western blot of animal tissues. You can see that there is increase in the DRP1 level of mitochondria in the animal tissue, rat, liver tissue. Cytochrome C level, it is also increasing the mitochondria. It is decreasing the mitochondria, and it shows that there is apoptosis of mitochondria. Here, basically, its level is increased. You can see in the triploid group, which is stored by the triploid plus MDB. This is the graphical abstract of my study. Here you can see that there is increase in ROS production and there is mitochondrial depolarization. Triploid has caused an increase in ATP production, mitochondrial fragmentation. There is energy deficit. MFN1 level is decreased. DRP1 level is increased. DRP1 level shows that there is mitochondrial fission, increase in mitochondrial fission, which causes release of mitochondrial, release of cytochrome C, which causes activation of caspase, and there is apoptosis and that apoptosis. This is the end from my side. If you have any question, then we ask. Thanks.
Assalamualaikum. Thank you very much for inviting me on the third International Brain and Biomedical Medicine Conference organized by Department of Physiology and Pharmaceutics at Government College University, Festival. I am Dr. Shukta Said from University of Veterinary and Animal Sciences of War. And the topic of my presentation is the synthesis of zinc oxide nanoparticles using Acanthus aspera leaf extract and evaluation of its antibacterial potential. First of all, introduction. Poultry industry played a very important role in economic development of Pakistan, and poultry meat consumption is progressively increasing. Worldwide reach 14.2 kilogram per capita per year, and mortality rate in poultry has been growing every year due to outbreak of several diseases. The major drawback in advancement of poultry industry, which lessen the prime production of dog and most of the microbial pathogens are salmonella and campylobacter. And microbial safety of poultry meat product is an important issue in context of increasing consumption and production. So these are the Salmonella gallinatum and Salmonella dietridis properties. Salmonella gallinatum is a broad-shaped gram-negative bacterium, and it contaminates eggs, thus making it potent infectious agent for humans. While the Salmonella dietridis is a low-shaped gram-negative facultative venom, and it transmitted via improper cooked meat, contaminated food, and water. Antibiotics Antimicrobial agents have long been used to treat different bronchial diseases and also reduce mortality associated with severe cases. However, they are becoming resistant against these antimicrobial agents. So, Salmonella varianarium present in chicken were resistant to doxycycline, polymyxin, streptomycin, and gentamicin. And Salmonella enteritis resistant to ampicillin, chloro, and picamidin. Nanoparticles are used as an alternative source, and the unregulated use of antibiotics provide the impulse for bacteria to become drug resistant. And nanotechnology is becoming as an alternative source against antibiotic resistant pathogens. And nanoparticles that develop antimicrobial approach for treating severe bacterial infection. And the basic size is less than 100 nanometers. So the green synthesis of nanoparticles is gaining importance day by day, and it has following properties like it is an expensive technique and it's time consuming. Sorry, and it has advantages over conventional method because conventional methods are expensive, time consuming, and use of chemical components like organic solvent as reducing agent. So various metal oxide nanoparticles like iron oxide, titanium oxide, copper oxide has been synthesized using plant source. So zinc oxide nanoparticles have been becoming area of interest due to their distinguished antimicrobial properties. And the zinc oxide is an inorganic compound, appears as white powder insoluble in water, and the activity of zinc oxide against various pathogens like Streptococcus aureus, Salmonella, Bacillus subtilis, and E. coli has been reported. Acanthus aspera is a species of plant in Amanthesia family, and its common name is carp or devil worship. Distributed throughout the world, and it habitated dry and warm places, and it has 1 to 2 meter in height and commonly found as a plant. So statement of problem of his research is the extensive use of antibiotic has raised concern about increased resistance of Salmonella species in poultry and cause serious health hazards and economic losses. Hypothesis: zinc oxide nanoparticles produced by leaf extract of Acanthus aspera have antimicrobial potential against poultry pathogens. So objective, there are two objectives of this study. First one is synthesis and characterization of zinc oxide nanoparticles, and then evaluation of antimicrobial activity against poultry pathogens. Material and methods. First of all, collection of sample plant, and it was procured from the Qadim campus, Lahore, from the Punjab University, and after collection, it was washed, so distilled water, and then dried in 160 degrees centigrade for 1 hour and dried in to make the powder. Then extract preparation. Extract are prepared with distilled water and then heat it for 25 to 30 minutes on heating mantle, and then filter the extract through the filter paper, and then obtain filtrate of stored. Then preparation of zinc oxide nanoparticles. 10 ml of leaf extract of Acanthus aspera, and it was mixed with 0.1 millimolar zinc acetate and sodium hydroxide, and then it was placed on magnetic stirrer for 4 hours. And after the 4 hours, sanctification was done, and the pellet was washed with distilled water and absolute ethanol. And then the obtained mixture is based on hot aroma to get the formation of zinc oxide nanoparticles. Then characterization of zinc oxide nanoparticles are done using following techniques like UV visible spectrophotometer, X-ray diffraction, SEM analysis, FTIR, and atomic force microscopy. Then evaluation of antibacterial activity of zinc oxide was done against Salmonella venereum and Salmonella enteritis. Method used for evaluation of antimicrobial activity was diffusion method and minimal inhibitory concentration. Well, diffusion method was done to check the antimicrobial activity of Acanthus aspera extract synthesizing oxide nanoparticles. Wells of 6 millimeter were made with fork border on a printed plate, and then plate surface was inoculated by swabbing microbial inoculum, and then 50 microliter of zinc oxide nanoparticles was poured, and then tetracycline and DMSO was used as positive and negative control respectively. Then it was incubated at 37 degrees centigrade for 48 hours, and then diameter of zinc zone of inhibition was measured. Then minimum inhibitory concentration of zinc oxide nanoparticles was obtained by using different concentration like 180, 60, 40, and 20 microgram per ml. And then antibiotic tetracycline was used as positive control, and DMSO was used as an active control, and then culture was incubated at 37 degrees centigrade for 24 hours, and MIC was calculated. Then statistical analysis was done using by mean and standard deviation on SPSS. These are the result of UV visible spectroscopy. As we can see that at 370 nanometer, zinc oxide nanoparticle show a peak. Then these are the result of X-ray diffraction analysis that was performed of synthesizing oxide nanoparticles. These are the atomic force microscopy image of the zinc oxide nanoparticles that basically show the surface morphology of the zinc oxide nanoparticles and radiography in blood. And these are the FTIR analysis that basically represent the presence of different functional groups present in the zinc oxide nanoparticles. And these are the SEM image scanning electron micrograph that basically represent the shape and size of the zinc oxide nanoparticles. These are the antimicrobial activity graph that basically synthesize that basically represent the against the Salmonella enteritis and Salmonella venereum, as it represents minimum heavy concentration was reported against 0.19 milligram against the Salmonella enteritis and greater than 0.39 milligram was reported against the Salmonella venereum. These are the images of well-defined method that represent the zone of inhibition. These are the MIC of nanoparticles for Salmonella venereum and enteritis that was 0.39 milligram and 0.195 milligram respectively. And when concluding, zinc oxide nanoparticles synthesized from Acanthus aspergillus extract has been proved as a better antibacterial agent against poultry pathogens. These are the things. And thank you very much.
Assalamualaikum. I'm Dr. Nabi Salman, and today I present my presentations organized by in organized by Department of Physiology and Department of Pharmaceutics and
बायोटेक्नोलॉजी वाला सेशन रह गया है कि वो हो गया पहले?
And the topic of my presentation is antibacterial, antioxidant, antidiabetic, bioactive secondary metabolites extracted from Urea ocean. And these are the aims and objectives contents of my topic. Objectives, scheme of study, method, results, conclusions, references. First of all is introduction. Secondary metabolites are bioactive organic compounds that inhibit the growth of pathogenic microorganisms. Bioactive secondary metabolites are natural products that show the antibacterial activity against different multi-drug resistant pathogenic bacteria. Bacteria produce various types of bioactive secondary metabolites. These metabolites have the potential to combat the variety of pathogenic microbes. The aims and objectives of my presentations: extraction of secondary metabolites from Urea ocean, determination of their bioactive potential, antibacterial, antioxidant, anti-inflammatory, antidiabetic. Identification of secondary metabolites present in Urea ocean by using spectrometry techniques. A scheme of study: growth of bacteria, production of bioactive secondary metabolites, extraction of bioactive metabolites, bioactivities of these metabolites, antibacterial, antioxidant, anti-inflammatory, antidiabetic. Identification of bioactive metabolites by using chromatographic techniques, GCMS and ESIMS. First is culture growth, and culture takes step and water path. Next, the culture was centrifuged, and the next step was filter and extraction of bioactive secondary metabolites, and solvent was evaporated by rotary evaporator. Results: Urea ocean antibacterial potential of extracted metabolites. The Urea ocean bioactive metabolites exhibited potential antibacterial effect against pathogenic bacterial strains Staphylococcus aureus, Escherichia coli, Bacillus cereus, and Serratia persistens. DPPH quenching acid, the highest inhibition activity, 83.87%, was observed at high concentration, 600 microgram per ml, and results indicated that the DPPH free radicals inhibited more effectively by increasing the concentration of extracted metabolites. Hydrogen peroxide quenching acid, the maximum effect of hydrogen peroxide, 91.93%, was observed at high concentration, 600 microgram per ml. Similarly, the anti-inflammatory activity, albumin denaturation acid, the maximum inhibition of protein denaturation, 87.67%, was reported at the maximum concentration of bioactive metabolites, 600 microgram per ml. And the percentage shows that the response was concentration of dose dependent manner. Similarly, the heat-induced hemolysis, the maximum inhibitory effect on heat-induced hemolysis, 81.39%, was observed at the highest concentration of extracted metabolites. Next, the alpha amylase inhibition, antidiabetic activity, the maximum inhibition, 90.97%, was observed at highest concentration of extracted metabolites, microgram per ml, and the dose response, the percentage increased by concentration dependent manner. The glucose uptake by heat, the maximum effect, 92.77%, was recorded at high concentration, 600 microgram per ml of extracted metabolites. Thirdly, the maximum inhibitory potential was observed at high concentration and of extracted metabolites. The percentage inhibition of pancreatic lipase was 76.73% at highest concentration. Identification of bioactive secondary metabolites by GCMS, various types of volatile organic metabolites were identified in GCMS spectroscopy techniques: nitrobenzene, beta phenoxyethyl alcohol, pentadecanoic acid, methyl esters, octadecanoic acid, 17 methyl esters, palmitic acid, methyl esters, palmitic acid, methyl ester, and olic acid, methyl esters, stearic acid, methyl esters, and similarly some other compounds, eicosine, docosine, and cyclodocosine ethyl was also present in GCMS volatile fraction. The nature of these compounds, some compounds are aromatic hydrocarbons, and most of the identified metabolites were fatty acids, methyl esters, like palmitic acids, olic acid, methyl esters, and pentadecanoic acids, methyl esters. Similarly, some long chain fatty acids esters were also identified in GCMS chromatogram, and some long chains alkenes like docosine was also present in GCMS chromatogram. And ESI identification of metabolites by ESIMS, some organic acids like succinic acid, glutamic acid, and capric acids were identified in GCMS chromatogram, and one drug, mebendol, anti-lipidemic drug, was identified in ESIMS technique. The ESIMS of succinic acid in negative mode. Similarly, the ESIMS of glutamic acid in active mode, and ESIMS of mebendol in inactive mode, and ESIMS of capric acid in active mode. So, to the best of our knowledge, identified bioactive secondary metabolites have not yet been reported from Pituria oceani Indian, and this work represents that the Pituria oceani bioactive secondary metabolites have the potential antibacterial, antioxidant, anti-inflammatory, and antibiotic potential. Further research is needed to clarify the precise mechanism of action and identification of most effective therapeutic application of bioactive secondary metabolites of Pituria oceani. So, these are the references, and thanks.
Assalamu alaikum. I hope you all are doing well. Thank you for joining today. My name is Hira Hamid. I have completed my MPhil in microbiology from the Islamic University of Bhagalpur. Currently, I am working as a research associate in Livestock and Dairy Development Department, Punjab, Pakistan. Today, I am here to present my research entitled as antimicrobial activity and characterization of zinc oxide and silver dog zinc oxide nanoparticles fabricated using Leucophyllum frucescens leaf extract in the 3rd International Brain and Biomedicines Conferences IBBC 2026. First of all, I will start from the introduction of nanoparticle. As we know that the zinc oxide nanoparticles have several beneficial properties, including low toxicity, biocompatibility, biodegradability, along with high photostability, thermodynamic stability, and high electrochemical coefficient coupling. Further, we know that green synthesis of nanoparticles, which I have used in my research, green synthesis of nanoparticles has gained a significant popularity in recent years of synthesizing doped zinc oxide nanoparticles, as it yields nanoparticles having superior morphological, phytochemical, photocatalytical, and electrochemical characteristics. Here is the methodology of my research. I took fresh Leucophyllum frucescens leaves, air dried, oven dried them at 100 degrees centigrade for 12 hours, and ground them into a fine powder. Then I prepared aqueous leaf extract. I use 110 gram of powder in 250 ml in distilled water at the conditions as 80 degrees centigrade for 30 minutes. Then filter it with the water and filter paper number 1 and storing the extract at 4 degrees centigrade. Then phytochemical screening was carried out to detect the flavonoids, saponins, alkaloids, carbohydrates, and these different tests. Zinc oxide nanoparticles were synthesized leaf extract into 200 ml of 1.1 molar zinc extract solution under continuous stirring for 2 hours. On precipitate was centrifuged at 8000 rpm for 20 minutes, then washed with distilled water and methanol, oven dried at 100 degrees centigrade for 12 hours, and falsified at 900 degrees centigrade to obtain pure zinc oxides. Silver dog oxide nanoparticles are similar, adding silver nitrate at different percentages like 3%, 5%, and 7% doping levels into the zinc acetate and leaf extract mixture. Synthesized nanoparticles, they were characterized using UV, visuality, FTIR, XRD, SEM, and TEM techniques to make sure about these nanoparticles. Antibacterial activity was evaluated against, I think this is right from here, anyhow, against Bacillus cereus, Salmonella typhi, E. coli, and gram positive Streptococcus aureus using different zinc doping concentrations. Here are the dramatical representation of the methodology: leaves, then dried made powder, solution preparation, filtration, extract, centrifugation, and then at the end of prepared nanoparticles. Here are some phytochemical analysis of L-frucescens extracts. Here are the results of these research. Figure 1 shows the UV visuality spectra. We can see that there are different pieces of zinc oxide nanoparticles, then silver doped at 3%, then silver doped at 5%, and then 7%. On the right side, figure 2 FTIR spectra. We can see its different peak values in centi per meter. Phytochemical screening of L-frucescens leaf extract confirmed the presence of flavonoids, saponins, phenolic compounds, and tannins. Ultraviolet visuality spectra showed a strong absorption peak around 277 nanometer, as I showed before. Here we can see the maximum peak at 270. Confirming successful formation of zinc oxide and silver doped zinc oxide nanoparticles. Further, we did FTIR analysis. This verified the involvement of plant biomolecular and functional groups in nanoparticle formation and stabilization. Then we did XTR analysis. It confirmed the crystalline nature and phase purity of synthesized zinc oxide and silver doped zinc oxide nanoparticles. SEM and TEM images revealed mostly spherical and polyhedral high agglomerated nanoparticles. here are the same images, and here are the 10 images. We can see the different values at different levels. Silver dog zinc oxide nanoparticles exhibited stronger antibacterial activity as we check the antibacterial activity against gram-positive and gram-negative bacteria. It shows that silver dog zinc oxide nanoparticle has stronger antibacterial activity than pure zinc oxide nanoparticle against R-tested pathogens. It means single zinc oxide nanoparticles have less antimicrobial activity as compared to silver dot. The highest antimicrobial activity was observed at 7% at the highest percentage silver doping, especially against Bacillus cereus and Streptococcus aureus. It means silver dog zinc oxide nanoparticles have more sensitivity against the gram-positive bacteria as compared to gram-negative bacteria. As I have showed this before to represent these images. Here is caption 10 images for synthesized zinc oxide nanoparticles with magnification of 0.5 micrometer than 100 nanometer than 5 different concentrations, different percentages. Here is antimicrobial activity of zinc oxide and silver dog zinc oxide nanoparticles. The graph shows the sensitivity bars of different bacteria. There is an inhibition zone against the active bacterial strains at a concentration of 50 microgram per ml. We prepared 1 ml solution by adding 50 microgram of silver dog zinc oxide nanoparticles. What we concluded: green synthesis zinc oxide nanoparticles and silver dog zinc oxide nanoparticles successfully produced using alpha-fructisens leaf extract as bio-reducing, stabilizing, and capping agent. Characterization we did XTR, PIR, and UV, which confirms high crystallinity, no impurities. Then SEM and TEM also shows SEM shows diverse nanoshapes in nanometer range. These nanoparticles should have then antibacterial potential. Silver dog zinc oxide nanoparticles exhibit superior activity. Whereas gram-positive and negative strains enable sustainable eco-friendly water purification. As we have noticed that it is more sensitive against gram-positive bacteria than gram-negative bacteria. Thank you so much. This is all from my side.
Assalamu alaikum. My name is Farzana Khalik and I am MP Scholar in Pharmacy. Today I am presenting my research under the title of Advanced Surplus-Based Solid Dispersion of Linear Enhancing Distribution and Pharmaceutical Performance for Improved Therapeutic Potential in Diabetes-Associated Complication. This study mainly focuses on improving the solubility and therapeutic effectiveness of linear, which is a BCS class 2 drug, and low solubility and hypermeability, which necessitate the need of improving the solubility by using an advanced solid dispersion technique. The first slide of my research will start with the challenges which the oral drug faced during the administration of the drug, like drug solubility, stability, patient population, first cause metabolism, systemic side effect, and drug permeability. Now, how to overcome these challenges? These can be overcome by using the technologies like particle size reduction, cyclodextrin usage, lipid-based systems, co-crystals, salt formation, polymorphs, and also some formulation strategies are also can be used to increase the solubility, like physical modification, chemical modification, which include the product formation and salt formation, and by reducing the pH of the formulation. Likewise, non-technological approaches are also used for increasing the bioavailabilities. These technologies, which are mentioned in these slides, involve the usage of lipid-based systems, which protects the drug from degradation by improving the resolution permeability. No doubt, all these technologies are very effective in increasing the solubility, but they have some limitations, like micronization have limited control on the size, shape, surface morphology, and electrostatical charges. Similarly, salt formation causes gastric discomfort, particle size, too much fine particle size causes the and solubility problems. Solid dispersion this technology can overcome all these limitations because it is the most commonly used, simplest, and efficient alternative to increasing the solubility. Sekiguchi and Bai propose this concept. It consists of two diverse components. One is the hydrophobic matrix, which is polymer, and another is the hydrophobic drug, which is either dispersed amorphous and resistant for particles within the matrix. It has several advantages like reduce particle size, increase porosity, increase wettability, conversion of the drug from the crystalline to amorphous forms, also offers many processing and recipients options. How we manufacture solid dispersion? There are several methods like heating methods, melting, co-grinding, co-precipitations, and separate drying, freeze drying, lyophilization, and electroscreening, co-supercritical fluids. I have used solvent evaporation method for making solid dispersion. The main objective of my research work was to increase the solubility and resolution of linear, which I have achieved by the sub-following specific objectives, like to evaluate the polymer effect on improving solubility and resolution, to characterize my formulation for FTIR, XRD, DS, and some studies. Next is the in vivo study to evaluate the therapeutic effectiveness, and the last one is the accelerated intermediate stability study to determine the aging effects. The methodology of my research work consists of preparing the solid dispersion, then testing for the physical characterization, solid state characterization, and in vivo study, then stability study. So the research will start with the first with the formation of formulation that I have prepared two formulation, binary and ternary, which binary involves the use of one carrier, that is the soluplus, which is the graft copolymer, and the ternary involves the use of soluplus, and next is the PEG, that is the polyethylene glycol 8000 grade. In these two formulation, I prepared for the co-formulation with the ratio 1, ratio 0.5, 1:1, 1:2, and 1:4. After then I prepared this formulation by using solvent preparation method. After the preparation of these formulation, I performed the test. The first test is the solubility test. The saturation solubility of the pure drug, as it is a class 2 drug, its solubility is very low, but the solubility of the solid dispersion is 18.69 milligram per ml fold, and it's the main goal of my study, which I achieved. Similarly, the next test is the in vitro drug release study. The binary solid dispersion, ternary solid dispersion graph shows that there is a dramatic shift in the release kinetics. The release of pure drug was very poor kinetic, but the release of solid dispersion was more than 40% within 5 minutes. I also compare my resolution profile with the marketed drug products, that is the average drug product, and the other is the Step 4 tablets. And then I observed the difference between these profiles. So the pure drug release only 10%, 10% in 5 minutes. Binary drug formulation shows 35% drug release. MRE shows 40%. Step 4 shows 40%, and the ternary solid dispersion release was more than 40% within the 5 minutes. Next step is the FTR study. No strong interaction was observed, as these graphs are shown, and the broadening and shifting of the key peaks was due to the hydrogen bonding between the polymer and the drug, which confirms that the drug is physically stable without any incompatibility. And next is the PXRD study. PXRD refractogram, as is shown, is the shown. The refractogram shows the decreased peak intensity intensity, showing the decreased causality and increased morphization. Next is summary analysis. DSC graph and TG graph shows. The DSC graph shows the distance of the sharp metastatic peak of the green upright, confirming that the drug is molecularly distinct and state. And the TG graph, no thermal degradation was observed, showing that the drug was remained stable during the manufacturing process, that is solvent evaporation process. And the next is the SEM study. The pure drug and binary ternary formulation shows the surface morphology. Surface morphology. The pure drug morphology was the interlike crystals and the binary and the ternary formulation morphology was very homogeneous. Next is the Nuvo study. The first test was the body weight test. Body weight, the diabetic group shows diabetic group shows weight loss, showing the catabolic state, while the treatment group shows the improved rate, showing the anabolic state. Next is the anti-diabetic as the group faster reduction in insulin of directly within. Next is the liver function test. The diabetic group shows hepatocellular injury and increased fatty enzyme and decreased protein level, and the treatment group shows in hepatoprotective effects and restoration of the hepatic biomarkers. And next is the renal function test. Renal function test, the diabetic group shows the renal dysfunction, while the treatment group shows renal improvement, showing the restoration of the renal biomarkers. Last one, the stability study. At accelerated and intermediate conditions, the drug content remains stable across all conditions at these conditions, and slight decrease in trend was observed over the time. So I concluded that the green solid dispersion can be successfully fabricated by solvent evaporation method, showing all the characteristics which are favorable for speed resolution and better absorption of the drug. The prospective aspect includes that the solid dispersion can also be fabricated by some advanced technologies like spray drying and automatic filling method. Next is the natural carriers like gums and silicates can also be used for improving the solubility of hydrophobic drugs. Then solid dispersion can also be formulated by 3D printing. Thank you very much. Thank you for your time and for this opportunity to share my work with IBBC. Confirms that the drug is clinically stable without any incompatibility. And next is the PXRD study. PXRD refractogram, as is shown, is as shown. The refractogram shows the decreased peak intensity, showing respected teachers and dear students, Assalamu alaikum. I hope you all are doing very well. This is Ina Shahzadi from Nutritional Sciences Department, and today I'll be presenting on liposomal nanocarriers, a novel therapeutic base targeting the gut-brain axis. This topic
focuses on how advanced drug delivery systems like liposomes can improve treatment strategies for complex neurological and metabolic disorders. So let's start with the introduction. So firstly, what is the gut-brain axis? The gut-brain axis is a two-way communication system between the brain and the gut. It involves the central nervous system, gut microbiota, and immune system. Recent research shows that this connection plays an important role in many conditions like neurological disorders, metabolic disorders, and certain mental health issues like depression. Although the gut-brain axis is an important target, but treating it is challenging because many drugs are unstable, get degraded in the GI tract, their absorption hinders in the small intestine. Also, their brain delivery is limited due to the biological barriers like blood-brain barrier. So, to overcome these challenges, there is need for more advanced and more efficient drug delivery systems. So here, why we choose liposomes and what are liposomes? Liposomes are nano-sized lipid vesicles made of a lipid bilayer. Its structure is similar to cell membranes. They can encapsulate both hydrophilic and lipophilic compounds. They are biocompatible and non-toxic. Worldwide, they are used for controlled and targeted delivery. Next is the mechanism of action, that how these liposomes work. They protect drugs from degradation, sensitive drugs like vitamin B12, enhance intestinal absorption, improve their bioavailability, cross biological barriers, and enable targeted delivery to the brain. Here is the visualization that how does this liposomal technology actually works.
To better understand how liposomes work in your body, I like to show you a short animation. When you swallow a capsule, it goes into your stomach and then leave your intestines. The acid in your stomach will cause the capsule to rapidly disintegrate and release its system helps create setup for an ideal absorption. However, with liposomes technology, the active ingredient has a formed into structures called liposomes, responsive to biolayers of molecules that help protect and transport the active ingredients so that they can more efficiently and ideally absorbed. And this is very interesting. Of course, liposomes are absorbed which are trained into the bloodstream. They are able to penetrate more rapidly into the tissues of your body than the antigens. In the final animation sequence, you'll see a comparison between the two processes. Liposomes which are formed in the body are something just body and resistance result in bioavailability of active ingredients.
Next is one of these important role of liposomes is in vitamin delivery. For example, vitamins like vitamin B12, as I mentioned earlier. Vitamin B12 is sensitive and get degraded in the GI tract. Its absorption is limited in the small intestine. So liposomes improve stability of vitamins, protect them from gastric degradation, enhance their absorption in the intestine, supports gut-brain signaling pathways, and is useful in nutrient-based therapies. Due to these advantages, liposomes have certain potential applications in several conditions. These include diabetic neuropathy, neuroinflammation, neurodegenerative disorders, depression, and metabolic disorders. Liposomes can also interact with the gut microbiota. They interact with gut microbiota by improving delivery of bioactive compounds. They influence the gut health and indirectly affect brain function. This enhances their overall therapeutic potential. Next are challenges. Despite their limitations, liposomes have certain challenges. The first challenge is stability issues. Liposomes can sometimes be unstable during storage. They may undergo leakage of the encapsulated drugs or fuse with each other, which reduces their effectiveness. Environmental factors like temperature and pH also affect their stability. Challenge number 2 is large-scale production. While liposomes can easily prepare in the lab, producing them consistently on an industrial scale is more difficult. Maintaining uniform size and quality is a major concern. Third challenge is post and formulation complexity. Liposome preparation involves expensive materials and advanced techniques. The formulation process can be complex, requiring careful optimization of components and conditions. This process increases the overall cost of production. Next are regulatory limitations. Regulatory approval is also a challenge. Since liposomes are advanced drug delivery systems, they must meet strict safety and efficacy standards. This can delay their clinical application and commercialization. So, while liposomes offer significant advantages, these challenges need to be addressed for their successful clinical translation. Next is conclusion. Conclusion from this presentation include liposomes offer a promising strategy for gut-brain axis modulation. It improves delivery of vitamins like vitamin B12 and neuroprotective agents, potential for treating complex disorders like diabetic neuropathy, neurodegenerative disorders, neuroinflammation, and mental health issues like depression. Future research needed for further clinical translation. Here are the references. Thank you.
So, Assalamu alaikum everyone. This is Muhammad Anas from the Faculty of Eastern Medicine, Hamdard University, and today I'm presenting my research topic, which is evaluation of the antidepressant activity and the safety profile of Becam, which is a polyherbal drug, and barley water, which is Maushai, in comparison with fluoxetine in a chronic, predictable, mild stress-induced depression model in Sprague-Dawley rats. So, before moving forward, we have to know that what is depression. So, according to DSM-5, depression is characterized by low or sad mood and anhedonia, lack of interest in pleasurable tasks, sleep disturbance, insomnia, or hypersomnia, or weight gain or weight loss, or any cognitive difficulty, or feeling of fatigue, for at least 2 weeks, and according to this temperament of cold and dry temperament. When we see the global prevalence of depression, it is very, very alarming that 5.7% of adults and 1.1 to 2.6% of children is suffering from depression nowadays, according to a study. And according to a recent study, this is very, very unfortunate that 8 to 15% of teenagers in 2026 is now suffering from depression. And these teenagers are early Gen Alphas and late Gen Zs. So 8 to 15% of them is suffering from depression nowadays. When you see prevalence of depression in Pakistan, then according to a study done in a specified population, in that specified population, 22% of that specified population was suffering from depression. When we see the economical burden of depression and its treatment strategies and its outcomes on Pakistan, it was 250.5 billion rupees in 2016, which has been increased to 617 billion Pakistani rupees in 2026, and it has been increasing day by day. So here is the aims and objective of my study. I have divided the aims and objective in two types. First is primary objective, which is acute and subacute toxicity evaluation of Be Calm and barley water, and these and the evaluation of their antidepressant potential. And the secondary objective is the antidepressant synergistic effect of barley water when it is co-administered with Be Calm and the fluoxetine. So here is the visuals of my test drug. Here is the barley water, which was prepared in the pharmacology lab of Faculty of Eastern Medicine, Hamdard University. And here is the visual of Becam, which is a powder drug filled in a capsule of a 500 mg capsule, which was prepared in pharmacy lab of Faculty of Eastern Medicine, Hamdard University. So here are the ingredients of these test drug. Barley water only contain Hordeum vulgare, which is simply jaw or barley, in addition with water. And Becam consists of 7 ingredients, which was Cascuta reflexa, and Nepeta hindustana, Polypodium vulgare, Rosa damascena, Terminalia jugula, Curcubitum mustata, and Delphinium denobatum. And these all herbs are traditionally has been using for psychiatric disorder and specifically for the depression. So here is the methodology of my study. This was the preclinical animal study in which we selected Spock Dolly rats, and the number of rats was 5 per group, and the chronic unpredictable mild stress protocol was administered for 35 days, and the permission of this study was given by Ethical Review Board of Hamdard University. So I have divided the methodology into 3 steps. The number 1 step is formulation of test drug. So for the formulation of test drug, we have selected and collected the required herb from the local market and then authenticated it in myself, and then the Department of Pharmacognosy Herbal Museum of Karachi University also authenticated it and preserved it and allotted the voucher number of each and every herb. Then after the authentication, the organoleptic evaluation was done, and then the preparation of compound drug, which is Aushayi or barley water, and the Becam was done. Then after that, the phytochemical analysis was done. Then after the phytochemical analysis, the toxicology of these drugs was done in 2 steps. The first step was acute toxicity, which was held by the OECD guidelines 423, and the subacute toxicity, which was held according to the OECD guideline 407. Then after the confirmation of its safety, the antidepressant toxicity evaluation was done. For this purpose, firstly, the induction of depression was done by a chronic unpredictable mild stress protocol for 35 days. Then after that, the confirmation of development of depression model was done by Saccharose preference test, open field test, and then the Foster test. Then after the confirmation of development of depression, the treatment was done for 35 days. Then after the treatment, the final antidepressant evaluation of the animals was done by Saccharose preference test, open field test, forcing test, and the tail suspension test. Here is the grouping of acute toxicity in Spock rats. For acute toxicity, single dose was given. The Becam was given in 3 different doses: 2,000 mg per kg, 3,000 mg per kg, and the 5,000 mg per kg. While the barley water was administered for 2,000 mg per kg. And for the subacute toxicity, the repeated dose for 28 days was administered, and Becam was given for 150 mg per kg, 300 mg per kg, and as well as the 600 mg per kg. And again, the barley water was given for 1,000 mg per kg. Here is the plan or grouping of animal for antidepressant evaluation. The animal was divided into 1, 2, 3, 4, 5, 6, 7, 8, 9. Here numbering is wrong. There are 9 groups. The first group served as control group, which was administered for distilled water, and the second group was the negative control group, which also administered for distilled water. But the difference was that the negative group has been induced with CUMS protocol. The third group was positive control, which was administered by fluoxetine 20 mg per kg, and the 4th, 5th, 6th, and 7th group was primary treatment group, which was administered for Becam in 3 different doses and the barley water. And the 6th and 7th group, or the 8th or 9th group, we can say, these are the secondary treatment group, and these were administered for a combination of barley water with fluoxetine and the combination of barley water with Becam. So here is the total study design or whole study design. Firstly, the animal was recruited from ICCPS Karachi University, and then acclimatized at the animal house of Hamdard University for 7 days. Then after the 7 days, the baseline Saccharose preference test was done. Then after the Saccharose preference test, the CUMS induction was done for 35 days. Then after 35 days of CUMS induction, the confirmation behavioral test, which was Saccharose preference test, open field test, and post test was done. Then after that, the treatment was done for again 35 days. Then after the 35 days of treatment, the final evaluation of antidepressant behaviors was done by Saccharose preference test, open field test, post test, and as well as the tail suspension test. Here is the CUMS inducers or stressors. 7 different stressors was given unpredictably to the animals, which was food deprivation for 12 hours, water deprivation for 12 hours, altered light and dark cycle for 24 hours, tail pinching of the animals for 3 minutes, case tilting for of the animals for 12 hours, wet bedding for 12 hours, and physical distance for 3 minutes. So here is the results of the study. The phytochemical analysis was revealed that there is no presence of steroids in our formulation except the ethanolic extract of Becam. And it is very interesting that the phytophenols was present in all the formulations of our test drugs, and which is very, very crucial for antidepressant potential in any herbal drug. So here is the result of safety profile or acute toxicity. The acute toxicity was done by observation of some behaviors, and all the behaviors of the animals in this all the group was normal and comparable to the control group. After that, the subacute toxicity, the hematological parameters and the hepatic and renal parameters was evaluated, and according to the result, all the values of these parameters were within normal physiological range, and there is no significant deviation from the control group, except here there is one value of these platelets of the calm mid dose which was very low in comparison to control group. So after that, histopathology was done of four we can say organs: brain, heart, kidney, and liver. Sorry. Okay, so there is no significant structural changes in histopathology in comparison to control group of the animals, which suggests that the intervention of our study, which was the calm and Maushi or barley water, did not cause any visible damage or alteration in cellular structure or tissue structure of any organ of any animal compared to the control group, and all the structures were intact. We can see neurocells, neurons, and blood vessels, and hair, muscle fibers, Bowman's capsule, glomerulus, central vein in hepatic side, and the sinusoids, and hepatic cells, and hepatic artery. All the structures was intact, and there is no any significant change in comparison to control. So after the confirmation of their safety, the antidepressant evaluation was done by Sacros preference test, Wilson test, appeal suspension test, and open field test. The result of the Sacros preference test revealed that there is the combination of Maushahir and fluoxetine revealed the most potent Sacros preference activity in the animals, which shows that the combination of fluoxetine and Maushahir decreased the anhedonia very potently in comparison to other groups and in comparison to control as well as the untreated COMS group. When we see the result of fluorescein test, we can see that the swimming time of Maushahir was very much increased, and the climbing time of BCAM mid dose was very increased. It shows that the Maushahir increased the you can say adrenaline in the brain of the animal, and BCAM mid dose of BCAM increases the serotonin in the animal's brain, and the immobility time of Maushahir group was very decreased in comparison to other group. All other groups shows positive and significant decrease, but in comparison to all the group, Maushahir showed the very much decrease. And when we see the secondary group or the combined or synergistic group, we can see that the combination of Maushahir with BCAM, Maushahir is water, and Maushahir with Ashwabat Amshal showed the significant activity in swimming time, as well as the Maushahir alone shows the significant activity in climbing time. When we see the result of tail suspension test, we can see that the high dose of BCAM showed the significant swimming time in comparison to fluoxetine group, comparable to fluoxetine group, and incomparable to control group. And after the fluoxetine, the immobility time was very much decreased in the BCAM low dose as well as the BCAM high dose in tail suspension test. Similarly, in the secondary group of the tail suspension test. So when we see the open field test, we have divided the open field test in 5 parameters, which is total distance covered, center time, rearing time, and the grooming count, and the defecation count. When we see the results, we see that the BCAM high dose increases the total distance covered in open field test of the animal, which shows that the high potency of BCAM can be administered in animals or the humans for their enzyme property or antidepressant property. Similarly, in all the cases. So the key finding of the whole study was combination treatment showed significant reduction in immobility time, improved Sacros preference, and locomotor activity in comparison to COMS control group. In fluorescein test, the immobility time was decreased. In tail suspension test, passive behavior was decreased. In open field test, locomotion was increased, and the Sacros preference test, the anhedonia was decreased. So this study concluded that the combination of barley water and BCAM and the combination of barley water and fluoxetine demonstrated significant enhanced in antidepressant activity in comparison to the alone administration of BCAM as well as the alone administration of barley water. So this validates the traditional or Unani concept of Ghazai Dawai and the Dawai Fazai. So we can say that we can administer barley water, which is a Ghazai Dawai, or you can say medicinal diet, can be administered with any drug which is fluoxetine or BCAM or any other drug, and it will work as a synergistic drug. So the safety was confirmed in acute and subacute toxicity, and the integrative therapeutic strategy was revealed in this study. This research highlights the potential of combining Unani nutrition-based therapy, which was barley water, with conventional pharmacotherapy, which was fluoxetine, for a holistic approach to depression management. So every study got some limitations. So our study have some limitations also. Our study was a clinical study which required human clinical trial for further validation, and the number of group number or sample size was very low, which was 5 animals per group, and the larger studies is recommended. This study was model and other animal model tested for this type of study. The mechanism of synergistic effect was not fully characterized at molecular level, but at the only behavioral level. So the future direction of the study can be the clinical trial in human subject with standardized Unani formulations can be done, and the molecular docking and pharmacokinetic study to elucidate the synergistic mechanisms can be done in future. And bioavailability studies and identification of active phytoconstituents can be identified in future, and the exploration of combination therapy for other neuropsychiatric disorder except depression can be done in the future. So here is the references of the study, and thank you so much for your time and your interest. I am Muhammad Anas. You can contact me anytime on my given emails. Thank you so much.
Assalamu alaikum. I'm very thankful to become part of this conference. My name is Fatma Qasim.